Okay, welcome back team. This is the second and final video for this week, and now we're going to actually really start to implement and apply all the tenets of scientific analysis that we learned in the first video. And of course, we're going to look at them through the lens of the assigned manuscript, which was the impact of diet in shaping the gut microbiota revealed by a comparative study in children from Europe and rural Africa. And so while we're going through this, I actually recommend you have your manuscript alongside whatever your video content player of choice is, and grab a pen and maybe some notebook paper and take some notes or write along the paper, whichever works best for you, as you learn some specifics that we want to point out here. As usual, this is not a comprehensive analysis of the paper. We're going to do some more in class, and even then we probably wouldn't hit it all. There's always something to unpack. So our primary learning outcome here for this video is to critically evaluate those section-specific components, that paper DNA that we've learned about, the formula of how things are put together, using our assigned manuscript. And I actually have a couple of outside references for examples later on. And then learn how to synthesize all of these new findings and associated significances to convey the information via written summaries for two different audiences, a scientific and a lay audience, as well as a graphical abstract. And this second bullet is really a free preview of part of the homework assignment. So it behooves you to stick around and listen in and learn how to look at some examples and then do your own as it pertains to this particular paper. And before we move forward, I always want to recommend that we are looking for both positive attributes in the paper as well as potential issues. We're not just going to implement the Festivus model of holiday airing of the grievances from Seinfeld, and I'm sure you don't know that reference, but that's okay. We want to not just bite at the article, but we also want to look for the good things too. This should help you not only read science, but also develop your own science. The best way to develop a new experiment, to ask a new question, is to read the literature itself first. One very, very wise scientist once told me that a day in the library, nowadays online, looking at articles, a day reading papers will save you a year or more at the lab bench, so that you know what's already been done, and so you know where to go next from where the literature is leaving off. So we're going to take a walk through this and point out some fantastic attributes as well as some potential pitfalls as well. So let's begin to evaluate. We're going to start our critical exploration of this manuscript, and I'm going to divide this up into three components. We're going to look at words and sentences used in the introductory and discussion materials. Then we're going to proceed towards the materials and methods. And finally, we're going to wrap it up with looking critically at the results. And again, we're not going to go through every single component, but this should give you a good idea of how to go forward. So here we are. First bit of this here, and again, have your paper and maybe a good cup of coffee next to you, or whatever other beverage of choice you might be enjoying right now. And let's look at the introduction and discussion. And so what I'm going to do is, on the left, give you a miniature version of the paper. Not that you should be able to see that, that's very small, but just to show you and highlight where I'm going to be looking at. So here we are on the first page, and I'm going to blow up that second paragraph down there. Okay, and it starts, "We do not yet completely understand." And the reason I'm pointing this out is because when you're first starting to think about the significance and of the work, and you want to make sure that the authors are conveying it to you correctly in the ways we described in the first video, this is where I kind of really start to do some digging. And I've animated a box over two keywords here: microbial ecology. And this is really, really at the heart of what this paper is about. We're going to compare two groups of people, and these are in 2 different parts of the world, on 2 different diets with other environmental factors that are different too, and we'll talk about that in a bit. And with the goal of characterizing those gut bacteria and looking for similarities and differences between those 2 populations. And so to characterize these things and show those differences really is a descriptive enterprise. You may notice that you don't see any mechanisms of action in this particular article, and that's okay. This is a very important first step to characterize these differences so that in the future directions, people can look in and see how a particular microbe may or may not be interacting with a dietary component or producing a particular metabolite like those short-chain fatty acids you saw later on. So this at the heart is a story about microbial ecology, and I like that they point that out to you right away. So that's a good thing. Let's move over to the second column, and I'm going to look at this paragraph right here. My arrow is blowing up this part right here. Western developed countries successfully ... This is the paragraph I'm going to look at, and we're still in the introductory materials, and this is still where the authors are setting up the story, and they really want to focus here on some of the human importance, some of that biological significance. Scientists always have to be good salesmen and saleswomen for their work. We have to advocate for what we're doing to get grant funding and also to go through the peer review process and publish papers. And so highlighting the potential biological implication for humanity is always a very important study to point out. And I'm going to focus in right here on this second sentence. At the same time, a rise in new diseases such as allergic, autoimmune disorders, and inflammatory bowel disease, both in adults and children, have been observed, etc. And this is a fantastic context for you to understand why we would want to even care about these gut microbiota species amongst different groups around the world. What I don't want you to do is make an overimportance on this particular line here. You might be tempted to think that you're about to learn something that will definitively associate with some of these disorders, like allergies, for example, or autoimmune disorders, or IBD. The authors don't and do a very nice job of not equalizing their findings with these at all. In fact, most of their results in their sentences lead to new hypotheses in their own words. We'll point that out later. But I don't want you, the reader, to think when you read this sentence that that's where we're headed. So we're walking a fine line here when we're selling our science and its potential significance and what we're really actually showing in this paper. Because I really want to remind you of an important indicator sentence that they've given you in their paper when they presented the study design, and we're going to come back to this later in that section. But that when they chose all the subjects in both of these areas, and in particularly the rural parts of Africa where diseases like malaria can be found, is that all these subjects presented as healthy. So therefore, we are not actually making any determination about irritable bowel disease or autoimmune disorders necessarily here in this paper, because all these subjects did present, as they said, as healthy specimens in this early part of their life. Whether or not these are indicators for these disorders later on as they age remain to be determined, and there's nothing here that we can gain for that definitively. So we're not going to overstate our importance. We are not going to, as we analyze this paper, gain anything more than what they're actually telling us. Moving to another part of the introduction, and we're going to this very last paragraph of the introduction on that second column, and I'll blow it up here for you. It says, in our study, we address 3 general questions. And they really do a fantastic job of laying them out for you in terms of what this study is going to show you. There is not an overstatement to be found. They are not suggesting they're going to give you a mechanism. They don't give you a mechanism. And you can really highlight words that indicate as much. So, for example, in the second of their general questions, it says, is there a possible correlation between bacterial diversity and diet? You may have looked at these figures and said, oh, fantastic. They've pointed out to me differences between these groups, and therefore these are important for health and disease. But we need to pause and slow down and remember that all these subjects were healthy and that we're not learning any mechanism. So a word of caution that all scientists must operate with, and that is remembering the difference between correlation and causation. There's nothing here that's showing causation. This is correlative at best. And to the credit of the authors, they're not suggesting anything more than that either. So we're okay with this. This is the first step of sort of thinking about how the story was set up and what the questions were. And if everything seems to be kosher in here, I think we are on a great path forward. Now let's go towards discussion material. And what I'm going to do is flip to that page where we really kind of get into it. And it's on the same page where you see in the right-hand column the materials and methods. And what I'm going to do is, and as indicated by my arrow here, focus in on that second paragraph of this first column for discussion purposes. And it starts with, altogether, our results indicate a correlation between polysaccharide degrading microbiota, ... Okay, so let's go ahead and digest. Sorry, that was a bad pun, I think. But I hope you understood what I meant. We're talking about microbes eating, and you may remember back in the biomath week, I called y'all food bags because you're nothing but walking microbes, and those microbes are eating, and so you are what you eat, right? So there, was the digest the paragraph joke anyway. Let's move forward. Okay, good. I'm going to start boxing to you statements containing words that indicate how you should think about these results. They're a correlation. Again, this is not causation. They potentially influence. We are not overstating our results here. If you had seen something like definitively influences or mechanistically influences instead of potentially influences, that should alert a red flag for you. But we don't see those types of conflations here. They're not suggesting causation at all. They're not potentially suggesting you should either. They continue by saying, now that we understand this, we can make some new hypotheses based on our results. And I really like that they did that. We can hypothesize that the microbes have co-evolved with the diet. And that's a very tempting hypothesis to make. And they could set up in the future some experiments to test this. And I bet you that group is planning to do that if they haven't already. If you skip down in the paragraph, it says substantial microbiota adaptation has probably accompanied. The authors here do a fantastic job of representing their data in this discussion section and what it might mean by saying it might mean or probably, and that future directions are definitely needed to dig deeper before we make any drastic conclusions about how this may or may not be influencing humans. I want to stay here on this theme, and we're going to move one paragraph down in the discussion to the paragraph that starts, our results suggest that diet has a dominant role over ... Let's start right at the top. Our results suggest something. They don't say that they show conclusively or definitively. They don't say prove. They say that they suggest something here. And that's important because we know there's other possible variables, and they do a great job of mentioning them. Ethnicity, sanitation, hygiene, etc. You can't rule based on how this work was done some of these out. And so they say suggest. And I remember this being such an important lesson for me as a graduate student, that my results suggested something. And I hinted at this in the previous video of how my advisor had to make sure I understood the difference between showing something in a test tube and then suggesting that I had the cure for a disease. No, they just suggested a particular observation was true in a test tube, and we had to leave it there. A mouse study is showing you what happens in a mouse. It's very, very arguable how applicable mice studies are to humans. We use them as models all the time, but there are significant differences, and that may explain a lot of the reason why beautiful studies in mice don't end up as clinical trials or successful therapies, or that there are human-specific differences that explain why these things may or may not work. If you keep going, they again say, we hypothesize then, based on the results. They could indicate. And so nothing seems out of order. Nothing seems overstated or exaggerated. I would say that there's a small exception to this as you finish out the discussion and look at the very, very last paper paragraph before you get to the methods. And that starts with the lessons learned from the BF Children's Microbiota. I want to point out the part that I think is a bit problematic or might lead people astray. And at the very last sentence, scientists typically try to really stretch and really think about what could potentially be the future of this work or its relevance, not necessarily that it is. And so it says that the worldwide diversity in the microbiome from ancient communities where gastrointestinal infections can make the difference between life and death represents a gold mine. Wow, okay. For studies aimed at elucidating the role of gut microbiota on the subtle balance between health and disease, and for the development of novel probiotics. Nowhere in here do you have any suggestion that of many of this. It could potentially be a gold mine. We may learn a lot, and we may elucidate, as it says here, what makes health and disease based on gut microbiota, and then potentially, if we have disease states, how something like a probiotic, which means a live bacteria that you could add to maybe a gut, in this case, could shift the species towards the ones that you're missing that we think are healthy. But that's a very, very far leap away from where we are right now in the world. The idea that there is such a thing as a, quote, healthy microbiota, a, quote, healthy grouping of bacteria that anybody should have or universally have, is a pipe dream right now to be a gold mine. And so this is where you might begin to think that there's a somewhat of an over salesmanship here at the end. And I really think the work could have just done it without this last bit right here after that comma where we just read. I don't think it's necessary whatsoever. If you didn't think of those things as you parse through the intro and the discussion, that's okay. This is, again, a skill that comes with time and practice. We're going to leave to now explore the second component of the paper anatomy, which is the overall experimental design and the materials and methods. We're going to look for limitations in them. Now, every scientist makes what I have in parentheses here, choices. There are a lot of variables in any experiment, especially in one like this where we have humans involved and all sorts of environmental factors to consider. So when you're setting up your experiment, as I mentioned in the first video, it's very important to define your parameters, establish your controls, so that you know when and where you can trust your data and make an important inference from your data to know what those results are suggesting towards what you're studying overall. The scientists performing the work at the bench after the design is completed make choices as well in terms of how much they're pipetting, or if they don't see a result, how to troubleshoot it. Maybe there was some user error, maybe there was some mechanical error. How do you figure that out and how do you move forward? We make choices. Those choices can limit us, and that's both good and bad. We have to live within the limitations of our choices. We have to live within the limitations of our models, even the mouse models that I just talked about a moment ago. So the most we can do is establish a very, very well-controlled study and perform well-controlled experiments. That's our job as a scientist, and we're going to learn how to do that later on in the semester. So let's move to the materials and methods, and I'm going to start right there with the very, very first portion of it. And the materials and methods on the second column of that last page there start with population enrollment. Something I love, fecal sample collection, and DNA extraction. So these are the very, very first steps in sort of conceptualizing this work, establishing how the poop was going to be collected, and of course the poop is where you find the gut microbes, and how the DNA was going to be extracted from those poop samples, the DNA of the microbes, so that they can be identified for the bacteria species that we learned about in this paper. Reading right off the top, it says, we enrolled 15 healthy children living in the rural village of Bull Pond district of Nanoro. And I'm going to pause right there, not because I'm struggling to read those words, but because of the number 15. You may have noticed that on the very, very first page of this paper, under the section marked Results and Discussion, the very first section is called Characterization of Dietary Habits of Children from this area. I'll read that first sentence to you. It says, in this study, we characterized the fecal microbiota of 14 healthy children from the Mazi ethnic group, BF for short. And then it goes on to say, and 15 from healthy European donors. So that right there is a discrepancy. This says in the methods 15 healthy children from this group, when actually it was 14. We know it was 14 based on some other evidence, and I'm going to go through that with you right here. And so hopefully this is just a typo, but your spidey senses might have been alerted if you looked at the methods and saw, that doesn't match. This is not correct. So hopefully this was just a typo. It says that you can look at table S1 for some more information. And I'm wondering if you actually clicked on the supplemental materials. Table S1, where the S stands for supplement, can be found in those supplemental materials. And so you've had the PDF of this through the Canvas platform. And if you scroll down there, there's a link. And if you click on that link, you can see all the supplemental materials. Those include an expanded methods and some tables that further explain the patient donors from both groups, as well as the diet and other environmental conditions. So let me show you that. Here is table S1, which I've copied from the supplement and put right here for you. And on the left column, you see ID, and so they've given an anonymous number to each donor from both groups, BF and EU, designating the different areas, Burkina Faso and Tuscany, Italy. So if you were worried about that discrepancy and you wanted to make sure that you had the right number, this is one place to find it. And so, as you can see, there are 14 BF samples listed in the chart and 15 from the European group. Therefore, we do think this, plus 29 being the number of total donors indicated further on in the text and in the figure legends, etc., we're okay assuming that it was just a typo there in the methods, and that everything was 14 BF and 15 EU samples in this particular study. What's more interesting to think about is the last two columns. In one, the route of delivery, and two, the number of months without antibiotics. And it's fantastic that you have this data here, but it certainly does pose a limitation, and they acknowledge it as such. when they're saying that we can't definitively go to causation, that there's other variables at play here potentially. And these are definitely two of them. So we know now, through the wonderful world of microbiology in primary literature, that the route of delivery affects the microbial composition that you get early in life, whether you're born naturally or via cesarean. And you do see a couple of instances here where children were born via cesarean. We know that breast milk and breastfeeding and the amount of breastfeeding one gets do change that. And so there were some differences between donors in that as well. And then, of course, antibiotics. And so this is a significant one in that the BF samples never were exposed to it, and the EU were, or at least for some period of time. And so these are very important limitations to make you think that there's potentially more than just diet at play when it comes to characterizing the microbiota from these groups. So there's a lot to dig in here into, and we might get into some more of these things in class. Doing great. Let's move on to the next section of the materials and methods. And again, we're not going to get into every single one, but I'm going to give you a few because we're again thinking about limitations based on choices. So this is 16S sequencing for rRNA gene amplicons. And so this is a very, very important choice. And in the world of sequencing, we are trying to determine identity. And there's two ways we could do that predominantly for microbes. One is to look at this 16S region. 16S is an indicator for something that all microbes have. And this particular region contains both conserved regions that are similar for every microbe, and then variable regions that make each microbe unique, and we could use to sequence and tell who's who. They're kind of like the barcoding regions of the 16S. And so that's what they mean by V in this sentence. V stands for variable, and that's what 16S is, where we're focusing on the 16S RNA region. And so I'm going to put those in boxes right here. The V5 and V6 hypervariable region. It just so happens that there are 9 variable regions within the 16S region, and they're only going to look at 2 of them, V5 and V6. Now, there's reason for that, and that's because a lot of the gut microbes fall within those 2 regions, 5 and 6, for their barcoding abilities. However, we're also excluding then bacteria that would be sequenced if we had probed, say, variable regions 1 and 2. We're not going to come up with those, and maybe those are actually key species that are determining differences between these 2 diet groups, but we just wouldn't recover them. Same if the key differences were actually in variable regions 7 or 8. We're not going to find those because the authors chose to focus on simply these 2 regions for their sequencing, V5 and V6. And so that's a limitation. It's based on good data, but it is nonetheless. Now, this paper was published in 2010, and fast forward now to today, and there are another way you could actually do the sequencing analysis for microbes, and particularly for bacteria, and that is to do a whole genome approach by something called shotgun sequencing. The main difference is between the 16S and the shotgun is that in the 16S approach, we're again targeting barcoded regions within one part of the bacteria chromosome. And in this case, it was even further down the rabbit hole, targeting V5 and V6, just those 2 regions. Shotgun sequencing chops up the whole genome and looks at all the DNA, and therefore would include all of those variable regions and other regions of the chromosome too, and with the added bonus of potentially giving us some predictors of gene function. So this is an advanced technology. Both of these have strengths and weaknesses associated with each, but you know what, that's for another class. But I just wanted to give you guys a heads up as to how the study was done, and that this represented an important choice. That then leads to some limitations in what you see. Now here's where that plays out a bit for you, and I'm going to actually, even though I'm still focusing on the methods, I want to look forward a little bit into the results, and really it's hard to separate the two. So the choice of methods and the limitations in design certainly affect the results. And here's that example then playing out for you, where the V5 and V6 sequencing decision affects the depth of what you see when you're looking at the composition of microbiota between the two groups. And so right here is figure 1, and I actually copied this over from the paper and put 1A side by side with 1B. And I'm going to indicate to you by showing this doubled arrow line that our resolution of sequencing only gets us to the phylum and genus levels here for the bacteria of interest. And so we don't actually get to species. I don't see anything beyond Prevotella, for example, as the genre, or Bacteroides or Roseberia. These are some of the example genre. The phylum level is the Bacteroides and the Firmicutes. So we can't see anything more depthful. For example, on the right in 1B, in the EU group, it says Bacteroides, the second one down after Allostipes. The Bacteroides genus contains a lot of species that may or may not inhabit your gut. And one of those is Bacteroides fragilis, which I always abbreviate in my teachings as BF, your best friend. It's just one of those bacteria that we know to now mechanistically train your immune system, keep inflammation low, and keep you healthy. You want to have that BF. We also know that not all BF are the same. And so there are also differences between genus, species, and then strain. So Bacteroides fragilis strain 1 or Bacteroides fragilis strain 2. And the difference between strains would be at the genetic level. And there's very important differences that potentially play out in composition of gut microbiota in humans. And so if all we get is to the depth of the genre here, and we don't even have the species, then we definitely don't know anything about the particular strains these people might have. And it's important because not all Bacteroides fragilis are the same. Come out to your skin. Not all Staph aureus are the same. There are different strains that live on your skin. In fact, in any given community of organisms in any given location, whether that's skin, gut, or soil, we can't assume that every single cell of a particular genus and species are exactly the same strain. So it's true that the shotgun approach would have gotten us a little bit closer, a little bit more depthful towards strains. But again, there's limitations, and so we're not just going to make that blanket statement just yet. Let's go down further then in the methods to the next section, quantifying and comparing diversity between BF and EU populations. And I really want to point this one out because it will be very consistent with things you've heard before in this class. Right off the bat, how do they analyze their data? Well, differences between populations have been analyzed using parametric ANOVA and something called non-parametric Kruskal-Wallis tests. Okay, ANOVA is something we've focused on, and I hope that's gotten your juices refreshed. There are a couple of quiz questions this week asking you to draw back on your material and your knowledge from that particular week. For example, you're starting to think about normal and non-normal distribution and whether or not this matters at all, right? Yeah, sure you are. Yeah. I can see you nodding. I know. That's good. So that's one component of it. Later on, they indicate that the non-parametric rank-driven Kruskal-Wallis test was preferred for some things. And it says, which performs well in absence of distributional assumptions. So we're going to explain some of these things in class, and there's actually a piece of your homework that asks you to look into this further, not just that quiz question. But these are important decisions. These are important choices based on the data they have, and why they actually chose to use these. And so what we're hoping now is that you're really starting to feel comfortable if we kind of think back to what this class has been to this point, as we're almost near the halfway point. So we've done data visualization, we've done organization of it, we then analyze that data via different statistical methods, and now we went to ANOVA. This reading of the paper and analyzing of the paper is also pulling in those lessons. As you can see, scientists making choices and publishing their work based on how their data is indicating they need to do that. Okay, so you're doing a great job, and we've crossed off 2 of the sections. Let's move to the third, and that would of course be the results. Again, we haven't hit them all, but that's going to be part of our class exercise this week. I'm going to actually go ahead and fast forward to figure 3. And I've got here the figure legend for the entire figure. Short chain fatty acid producing bacteria could help to prevent establishment of some potentially pathogenic intestinal bacteria. And I have part A illustrated for you here. And so what you see is on the y-axis, short-chain fatty acids in fecal samples given to you as a particular concentration, micromolar per gram. You have a black set of bars and a gray set of bars where those colors differentiate the diet groups, black being the BF and gray being the EU. And then you can see comparisons for, on the x-axis, total short-chain fatty acids, or each of them the 4 individual ones that follow: acetic, propionic, butyric, and valeric. As you can tell by the asterisks, I actually don't know. I always biff on that pluralization of asterisks. Say it how you like. There's either 3 or 2, but in any case, you can see at the bottom that the p-values based on one-tailed student t-tests all fall in significant ranges, where in this case you've got 2 or 3 in figure 3a are all either less than or equal to 0.01 or 0.001, depending on if it's 2 or 3 respectively. That's interesting. One of the things I've taught you in the first video to think about is looking for sentences or statements in the results that don't quite match the actual figures. This happens to be one of those times. And so here is the text from the page that follows the figure where the graph is actually being referenced. And so I'm going to box this part right here. It says, whereas acetic and valeric acids were comparable in both groups. Let's read that whole sentence. In particular, propionic acid and butyric acids are nearly 4 times more abundant in BF than in EU fecal samples, whereas acetic and valeric acids were comparable in both groups. That word comparable suggests that they're the same and that they wouldn't be significantly different. However, if you look at the graph for both acetic and valeric, you do see that the diet groups are differentiated by asterisks that indicate statistical significance. So this actually doesn't quite line up, and this would be a experimental critique related to the results as written. As a quick aside, related to bar graphs and showing data in that representation, and we've talked about data visualization, I'm giving you a reference here at the bottom. That's the DOI number for a paper. And this is a really, really nice paper that actually looked at how particular probiotics protected against Vibrio cholera infection in the gut. And the reason I like this presentation better than bar graphs, which they could have absolutely given you, is that you see all the individual data points and their distribution within each group. So on the x-axis, you see V, which is how much Vibrio you'd get. And the second group, V plus L, is how much Vibrio you'd get if Vibrio and a probiotic marked L were added together. So there's obviously a significant difference between the blue and red groupings. The line indicates the average for each of these sets. And so not only can you count the number of data points in each, so 1, 2, 3, 4, 5, 6, 7 for blue, and 1, 2, 3, 4, 5, 6, 7 for red, that's consistent. Our n is the same. And of course, looking at sample size and consistency between sample size is certainly something we're thinking about, as mentioned in video 1. This is really nice because it gives you the entire distribution, and you can see, for example, where some data points overlap with other data sets. So look at those two red ones overlapping with some of the, including the average values in the Vibrio group on the left, or the blue ones. And that's nice to see sort of that biological variation and have an appreciation for that. It's not simply all or one, all or nothing. You have some data points that do indeed overlap between groups for especially things like, in this case, a mouse study where each of those circles is a different mouse. There's a lot of biological variation to consider. So it's nice to see it represented. So here's an example where it's hard to tell whether or not the sentence as written and the results as given are in alignment with each other. Where we're going to look is right above figure 3. We're going to go to the bottom of that left column, just above it. And I'm going to blow that up for you here as indicated by the arrow. We're going to focus in on the section that says microbial richness and biodiversity. So as we then compared the richness as estimated by the Cow1 index and the biodiversity assessed by a non-parametric Shannon index for the two BF and EU groups. In our calculations, we took into account different OTU distances. Fantastic. And then it says, using the non-parametric Kruskal-Wallis test for comparisons, we found significant differences, I'm going to blow that up for you, in both richness and biodiversity between these two groups. The question is that actually what the data does show you? When you look at this S6, it's a bit hard to really see what's happening. I'm not going to put the box plots from figure S3 that do show the Shannon indices generated from non-parametric analysis. The thing that's confusing is that when you look at this table, which doesn't reference in the text NP Shannon indexes, they just refer to those in figure S3. You see in the last 3 columns NP Shannons with p-values that are not significant. So it's very hard to reconcile then sort of this discrepancy between those two things. And sometimes you see that, and there's valid reasons for this, but we can't really make that determination given the 2 different presentations of those data sets. All right, so let's take a deep breath. That concludes the analysis of this particular paper portion of the video. We didn't analyze every bit. We've left some for class, and we've also left some for you to think about in your written work for the homework component, and that's what we're going to talk about now. Our main hope is that at this point you feel much more confident with the article and have learned some ways to parse through a scientific manuscript to make it feel a little more accessible and not as scary, which it may have when you first picked up this article. I remember feeling that way, and I still do. Reading science is challenging, and so learning the formula for how to think about it and how to make your own determination of what to think about these results is so important. It's really to me, one of the most important things you could do with your bachelor's degree is learn how to be a critical thinker in all contexts, and of course this being one of them. And so you can kind of ask yourself, did you enjoy that process? Did you enjoy sleuthing for clues through an article, seeing how the scientific method was employed, how all of the elements of experimental design came to be? Because if so, you should approach one of your instructors and we can discuss with you some opportunities that you might be able to take part in during your undergraduate, or even point you towards some postgraduate opportunities, whether those be graduate programs or jobs, so that you can use these skills and continue to learn more about them to see if this is something you might want to do for your career. And having said that, want to give you some overview on this week's writing assignments for both the homework and the discussion we're going to have in class. And this is kind of then going towards now putting it back together. We spent some time looking at all the puzzle pieces up until this point. And now we want to put those puzzle pieces together. We're going to synthesize all this together by summation. And we're going to do this in 3 different ways. You've already now in class been tasked with writing the abstract for the article. And that was a challenging thing, no doubt. So that was one form. And so we're going to build on that. But we're going to ask you to do these 3 things. The first is to create a short, abbreviated, written synopsis, let's say, of the whole paper, and we want you to do so for a scientific audience. The goal here is to, in just a couple of short sentences, in your own words, be able to convey the main findings and the main significance. So the main findings and the main significance for this article, as if you were writing to fellow scientists. Learning how to condense an entire story into a couple of short sentences is a very, very important skill that all of us scientists must learn. And it applies to many situations that you might not be thinking about. Like, for example, someone might ask you at some point, well, what do you study when you're doing undergraduate research? And you don't have a lot of time and you don't want to go on and on. So learning how to briefly describe sort of like an elevator pitch version of your work is key for a lot of different places. And so we're going to do this in the first part, and there's more directions for this on the Canvas platform. The second component is taking the scientific audience synthesis you did, conveying those main findings and biological significance of the story again, but in this instance for a lay audience. And this is really challenging. This might sound like it's the easy part of the assignment, but actually this is really hard. And so we're going to give you a link online to a database of the most commonly used words. I think it's the 1,000 most commonly used words in the English language. And you're going to copy paste your writing in here and check to see if you have a version that fits within these words. So we're going to have a very strict word category so that you are learning how to discuss the findings of the paper with someone who might not be a scientist, might not be a biologist. I think this is one of the most important jobs of a biologist, and that being to share your knowledge with people who might not be as lucky as you to be earning a bachelor's degree in this field, and so that they understand the world around them as much as you do at such an advanced level. This requires us to change the way we speak a little bit. We've spent so much time in this class learning the scientific language for reading and writing, and that's great. That's going to be your job and your career. But it's a very different thing then, of course, from how we would communicate this maybe with friends or family who just aren't in this field of study. And look how hard it was to learn all this. So people who haven't then are relying on you to provide them this information. And just as a quick little related note, if we don't, as biologists and scientists more broadly, talk to the public about what we're doing and why it's important, then we're going to continue to struggle with a lot of things like antibiotic resistance and people understanding and appreciating why and how we can tackle this problem. The majority of scientific funding to put instruments and people in labs and do research is funded by taxpayers, the general public. And so we have to convince them that this is important, show them why they should care, and help them realize what we're doing so that they do have an appreciation at that level. So we're going to have you write a lay audience summary, and that actually is going to be harder than you think. And I actually very, very much encourage you this week when you're doing this is to write the scientific summary and write the lay audience summary, and then read them to your roommates, your family, your friends, your BFs, your GFs, all the people around you, and get some feedback and see what they say. See how much they understand of the scientific one, and then if they totally get it when you flip over to the lay one, that would be fantastic. The third way we're going to have you synthesize is by making what's called a graphical abstract, and I'm going to explain that in the next slide. And this is actually becoming a more and more commonly used format for journals. They're going to publish a scientific manuscript, and alongside it is going to be a graphical summary of the article. So this is to summarize the article in a visual way. And basically what you can think about is taking your abstract that you wrote and putting it down, and then side by side bringing it to life through graphics, and we're gonna give you some options for how to do this. You can hand draw it in color, you could use a Adobe Illustrator program, you could use Microsoft PowerPoint. Examples are coming here in just the next slide, but this is a really effective way. I'm a visual learner. I usually don't understand anything until I see it drawn, or especially when we're talking about mechanisms, having this lead to this with an arrow, and then how does this get to the big picture. That's what we're gonna have you guys do, each of these three things. So I'm gonna go in the next slide here about more information for graphical abstracts. Okay, so behold an example, and again I've given you the reference down at the bottom with the DOI value, of a graphical abstract as a means of scientific synthesis and communication. And one of the reasons we want to have you do all of these different components of the scientific process throughout this class, including this one, is so that you begin to realize strengths you may have and weaknesses. And we want you to go follow up on your strengths and as indicators of potential career paths that any of these individual skill sets involved in science might be exciting for you to try. And then for the weaknesses, to help you identify areas of need of improvement, and potentially ways of you filtering out things that you don't enjoy doing as ways of eliminating potential career paths. Science is not a one-size-fits-all career. It's not all lab bench. It's not all healthcare. There are so many things you could do. This actually plays right into something called science illustration. And I love this because many journals now, and we learned about peer review and journal publication in the last video, many of these journals are now requiring scientists to publish a graphical representation of their story, a graphical synthesis. And some journals even have staff now to help with this. And you've seen some good graphical representations in textbooks and through Google image searching of a concept. In fact, as a little side tip, the journal Nature Reviews, which is a review article, not a primary literature article, but those review articles come with several beautiful graphical synthesis figures of different mechanisms and concepts. And so as a student, one of the little study tips I recommend is when you get stuck on a concept, go to Google, enter that concept, and then type in the word Nature Reviews after it. So concept and nature and reviews, and then do an image search and see if you can get a really nice synthesis summary figure to help your studying. Because I was a visual learner and I actually really enjoyed visually presenting data, and you might be too. And you might not have even known about these tools. Here is one such version. This actually came in a complex, super challenging article that was very mechanistic in nature from the journal Cell, Host, and Microbe. And this was published earlier this year. And it shows you sort of the whole mechanism. This is actually a story about the skin and the bacteria you're going to find on the skin, and how vitamin A has a big role in sort of how the resident microbiota, not of the gut, but of the skin, sort of takes shape, and then how it may or may not lead to pathogenic infections, in this case of streptococcus. It was a really nice story. This is such a really nice way, when you follow the arrows, of learning about a story. And in fact, every nice graphical abstract comes with a series of descriptive bullets that go with the story. And I'm not going to read through these here, but the important thing is that you could essentially draw a line from any of those bulleted words over to any of the lines and pictures on the left, and paint a complete picture of the individual findings on the right and how they led to the big picture on the left. And that's just such a nice way to put things together. So again, you see each of the individual findings, each of the pieces of the mechanism described out on the right, and then put together on the left. And it's definitely okay if you're not following exactly what those findings are. That's really not the point. The point is to show you how this looks and how it works, as you are going to be asked to create a graphical abstract for this particular paper, where we want you to create a summative cartoon or drawing, and then create some summary bullets or highlights that go with it to explain your drawing. And more depthful directions will be found for this within the assignment section here. So this second example, and the last one I'm going to give you, is also from the journal Cell, Host, and Microbe, and you can again see the reference down there, and this was published very recently here. And it involves the bacterial microbiota, and you see a person doubled over in pain, and then a blow up there of their intestine, and you can see this is also a story about regional differences, here focusing on Bangladesh. And within there, you see that these two bacteria are being what's called differentially affected by bacteriophages. And that is quite okay if you have no idea what I just said. I wanted to show you this because of the inherent similarities to what we're doing, but also to show you that sometimes graphical abstracts are accompanied by longer summaries, which almost look like a written abstract. And so again, the goal of your assignment is going to be to put your written abstract that you wrote on a screen or paper, and then make it come to life in a drawing form, and then for you to create summary bullets for the synthesis part, like in the previous example on the last slide. You can hand draw, you can color and hand draw, you could use PowerPoint, you could use Adobe Photoshop, you can let your imagination and creativity run wild. You can draw and take a picture and upload it for the assignment. Whatever you want to do works for us. Or create little cartoons in, again, any electronic software of your choice. This actually concludes the video for the depthful analysis of the article that was assigned to you this week. And I hope this helped sort of get you thinking, get your juices flowing of where to look and how to look, and then to be able to go find some examples of your own as you kind of sleuth through the clues. And we'll see you in class soon. Thanks again for your continued attention and participation.